Still not? Okay, now it's working. So we'll be moving out of the nucleus in this last talk, which you can kind of consider as a warm-up for the session after lunch, which will be on protein homeostasis. Because what I'll be telling you about today is how important translation speed and specifically codon translation rates are for maintaining your proteome in a healthy state, and how these translation rates are modulated by tRNA modifications. So proteins begin to fold as they emerge from translating ribosomes, and it's becoming increasingly clear that the speed of translation can actually affect the probability that they will reach their native state. And during each round of elongation, the ribosome has to select a tRNA that matches the codon and its A site from a pool of very, very similar molecules in the cytoplasm. And the nature of codon-anticodon interactions actually can influence the speed of decoding, because when a matching pair is formed, this actually significantly accelerates the downstream formation of the peptide bond. And a matching pair is recognized by Watson-Crick base pairing geometry in the decoding center of the ribosome. But what's interesting is that at the third position of the codon, there are some non-standard or so-called wobble base pairs that are allowed that actually give the chance of some tRNA molecules to decode more than one codon. And what's remarkable about the wobble nucleoside in tRNA, which is at position 34, is that in all kingdoms of life, it very frequently carries chemical modifications to the base. And if this is a wobble uridine, or U34, this is almost universally modified. And the genes that are required for this in eukaryotes are very highly conserved. And actually, a whole lot of genetic information is dedicated to modifying wobble uridines. And at the 5-carbon of the base, the 6-subunit elongated complex adds a methoxycarbonyl methyl or a carbamyl methyl group, and this occurs in 11 tRNA isoacceptors in the cytosol. And 3 of those are even more special because another 4 genes are needed to add a sulfur group at the 2-carbon. This is the ERM1 sulfur relay pathway. And these tRNAs, which decode lysine, glutamine, and glutamic acid, are thiolated at this position in all organisms. So actually, this modification forms part of the core translational apparatus of extant cells. So why would you bother to do all of this? Well, actually, it's not important for tRNA stability or amino acylation, but when you knock out one of these enzymes, it actually has profound consequences for cellular and organismal fitness. And what happens in yeast is that it actually sensitizes cells to a variety of environmental stresses. And in C. elegans, it causes neurological and developmental defects. And wobble uridine modification becomes more and more important with increasing developmental complexity, because a knockout of these enzymes is actually embryonic lethal in Drosophila and mice. And mutations in the human orthologs have been associated with neurodevelopmental disorders. So what I really wanted to know was whether there's actually a common molecular denominator for all these very diverse phenotypes in these different organisms, and whether this can actually relate to codon translation speed. So to find out how wobble uridine modifications maximize cellular and organismal fitness, I wanted to find out whether they actually modulate codon translation rates in vivo. And to do this, I used ribosome profiling, which comprises the deep sequencing of mRNA fragments that are about 30 nucleotides and are protected from nuclease digestion by bound ribosomes. And this method allows you to generate a quantitative snapshot of translation in living cells. And the codon resolution that it provides actually allows you to look at codon-specific translation defects. And so I use this to compare codon level occupancy in a wild-type yeast to a strain that lacks wobble uridine thiolation, one that is defective in 5-carbon modifications at this base, and a strain where all wobble uridines were completely unmodified. And my expectation was that since slower translation would increase the probability that you would capture a ribosome footprint by sequencing, if the hypomodified tRNAs are worse at actually decoding their cognate codons, this would manifest as increased ribosome density at these particular codons, especially when they're in the A site. And so when I compared A site codon occupancy in the 3 mutants to that in the wild type, what I noticed was that actually ablating the wobble uridine modifications had really strikingly different effects in different tRNAs. So there were 2 codons that are read by the thiolated TK and TQ that were reproducibly enriched in all 3 mutants, suggesting that they're really decoded much more slowly when the wobble uridines in these tRNAs are not modified. But there was very little difference in codon occupancy at all other positions decoded by hypomodified tRNAs, suggesting that decreased translation rates are not a universal consequence of modification loss. And actually, this fits really nicely with genetic data that we've been puzzled by for a very long time, which actually shows that the stress sensitivity of the wobble uridine modification deficient cells is complemented only by overexpressing the TK and TQ, still in a hypomodified version, with an additional minor contribution of the TE isoacceptor. And we could tie this complementation to relieved ribosome pausing, because when we overexpress these tRNAs in a mutant background, what we see is that the ribosome pausing at their cognate codons is relieved to a very large extent. So what we saw is that modifying wobble uridines is important for maintaining proper decoding speed at a subset of codons, and that this somehow leads to maintaining the ability of cells to respond to stress. But how can we tie these 2 very different things together? So what we thought is the best way is to essentially ask cells, how do they deal with the consequences of slower decoding? And to do this, we looked at gene expression changes in the mutant strain compared to the wild type, just during normal growth. And what was really remarkable was that there were many, many genes that were differentially expressed in the mutant, and the ones that were induced were significantly enriched for components of the protein degradation and refolding machinery, and in particular for heat shock proteins and the ubiquitin proteasome system. And this was not a spurious decision to upregulate these pathways, because if we knock out RPN4, which is the transcription factor in yeast that maintains high proteasome levels, this is really detrimental for growth of cells with wobble uridine modification defects. So this kind of suggested that there is some sort of proteotoxic stress going on. So we thought, well, are there actually some aberrant misfolded aggregated proteins in these cells? So we took our wild type and the mutant, and we just let them grow to exponential phase in rich medium, which is the best possible conditions that they could encounter. And we did a quantitative isolation of aggregated proteins from these cells. And what we could see is that in a wild type situation, there is essentially nothing that aggregates under these conditions, but there are a bunch of proteins that are coming out of solution in our mutant strains. And what was very interesting to see is that these proteins were actually kind of the usual suspects. So proteins that are known to be aggregation prone upon folding perturbations in vivo, because the pattern of aggregation that we see in our mutant is very, very similar to the one of a strain that lacks the major ribosome-associated chaperone, SSB. And so with mass spec, we were able to confirm this overlap, and we actually saw that many of the proteins aggregating were known aggregation-prone metastable proteins, like components of the ribosome and its assembly factors, and also a number of kinases that are reliant on chaperones for folding. And so what we think might account for this is that some proteins that misfold upon translation defects might sequester or otherwise impair critical proteostasis regulators and diminish the capability of cells to keep proteins in a folded state. And we could tie this protein aggregation to inefficient decoding, because when we used our trick to restore decoding speed by overexpressing these 3 tRNAs, we could actually alleviate the aggregate burden in the mutant strains to a very, very large extent. And we also saw that the gene expression changes that were a signature of the mutants were actually reverting back to very close to wild type levels, so we didn't need higher levels of protein quality control. So what we saw in yeast was that inefficient decoding through the unmodified tRNAs leads to a breakdown in protein homeostasis. But as I told you in the beginning, there were very diverse phenotypes that we associate with the loss of modifications in different organisms. So we wanted to confirm our key observations in a metazoan, and we used C. elegans for that. And we first looked at codon occupancy in a wild-type nematode compared to a thiolation-deficient mutant. And what we saw also here is that there was a remarkable increase in the codons read by the hypomodified tRNAs within the A site of the ribosome footprints from the mutant, confirming that the requirement for these modifications for efficient decoding speed is conserved in metazoans. And to probe for the folding environment in these nematodes, we've tracked a folding sensor, which is composed of a stretch of 30 to 35 glutamine residues fused to YFP. And the sensor is widely used in the worm community, and because it actually misfolds in the presence of other misfolded proteins. And so what we saw is that our mutant worms had a much higher aggregate burden than those that were age-matched wild-type controls, actually. And the worms with unmodified tRNAs were also expressing higher levels of the cytosolic chaperones. All of this allowing us to confirm that protein homeostasis defects are a common consequence of wobble uridine modification loss. So by actually exploring the consequences of ablating modifications in the tRNA anticodon, we were actually able to show for the first time that a slowdown at a couple of codons only, when it's not programmed in a cell, is sufficient to trigger proteotoxic stress and the aggregation of many, many endogenous proteins. And cells and animals are normally able to cope with a certain amount of increased aggregation by increasing their quality control systems. But when these systems become overloaded, whether it's in the, by external stress or during aging, the collapse of homeostasis then very likely underlies all the defects in growth and homeostasis that we see associating with wobble uridine modification loss in different organisms. So with this, I'd like to end by thanking my postdoctoral mentor, Sebastian Leidl at the MPI. I'm very grateful also to EMBO for funding this project, and to a number of people that made sure our sequencer is running at our mass spectrometry facility, which was a fantastic help in identifying the aggregates. And I'll be happy to answer any questions. Thanks for your attention.
Okay.
So you nicely showed that you can relieve the phenotype by tRNA overexpression, but can you also relieve it by overexpression of the chaperones?
So we've tried that. What's a little bit difficult, so we've tried with co-translational chaperones, and the thing is that they're actually co-regulated with the ribosome in yeast, and the ribosomal proteins are downregulated as a common stress response, essentially. So we're not really able to achieve a very high overexpression. We need to work a little bit harder on that.
Pedro.
Hi. So my question is related to all this. Do you know whether these wobble-modified codons are enriched in certain classes of chaperones? Did you look into that?
Whether they're enriched in the chaperones.
So like, for example, SSB1 and 2,
right.
Are the levels
Right.
of these proteins actually lower in
so
these cells? Because
yeah,
the
so as I alluded to, the entire ribosomal pathway is downregulated in all of our mutants already at the transcription level, which is again part of the common environmental stress response. So we see also at the transcriptional level downregulation of SSB1 and SSB2. And at the protein level, it seems to be very close to the one that we see at the transcriptional level. So we think that most of the changes are actually occurring already at the level of transcription.
If you enrich on the aggregate, and you do mass spec, do you see more mutations in the peptides?
Yes, so that's actually...
Or you see peptides missing?
So that is actually one of the things that I would like to go on with. So what we think right now, what we're looking at in these aggregates is kind of the tip of the iceberg, because we haven't perturbed anything, so there's a lot of turnover going on. We have been having some trouble to find specific mutations, because in mass spec this is a little bit more difficult than in sequencing, so you need to kind of build a database with peptides with certain mutations, and so far we haven't really been able to identify any, but it could be a sensitivity issue as well, so it's one of the things we want to look at.
My question draws on the last question. So just to clarify, when you say mutations, what you mean is misincorporation of amino acids?
I think in this case,
Right.
yes.
So in your narrative, you emphasized the slowing down of elongation. But wouldn't it also stand to reason that the slowing down might allow the incorporation of
Absolutely.
incorrect
Absolutely.
tRNAs.
Yeah.
So it's not really about slowing, it's about misincorporation.
It could be either of the two, depending on the context. I think it's going to be. I think it's going to happen in different proteins. Different things are going to be important. The fact that we can bring up the levels of these hypermodified tRNAs in complement would rather argue against massive misincorporation, because then you might think that you might even increase it in that case when you flood the system.
But aren't you suppressing, by doing that, you're simply suppressing the competition for
Yes.
non-cognate tRNAs
Yes.
at those. So that explanation is entirely coherent
Yeah.
with misincorporation. And may I also ask, your story isn't, the notion of translation, of attenuated elongation, there's a little cottage industry that would have you believe that by slowing down elongation, actually you can promote proteostasis.
Absolutely.
I believe there are examples where mutations in CFTR have been attenuated, the consequences have been attenuated by slowing down the rate of protein synthesis. So how do you reconcile?
So in this case, I would rather say that we're moving away from the optimal speed of translation for these codons in this particular context. So by speeding up or slowing down in an unplanned manner, you can in both cases trigger misfolding, I think. So this would be my explanation, that I know that a slowdown is a good thing in most people's heads, but in this case we're talking about slowdown that was not programmed. So these genes have not evolved in a way that has been adapted to a slower speed of translation of these particular codons. So I would rationalize it like that.
Okay, I think we have to. Well, Fatima, you want one question? Okay.
It's just a curiosity. Are there proteins, that contain, so RNAs that contain, are enriched in these codons recognized by these tRNAs, and if so, what is the encoded product?
So for the moment, we've tried to kind of stay away from bioinformatically predicting what would be affected or not, because you don't necessarily need a stretch of those codons to induce misfolding, because often even a single synonymous codon change is going to lead to protein misfolding. So we wanted to take an as unbiased approach as possible from the beginning. Of course, now we're trying to dissect a little bit more in detail whether these kind of things would matter or not.
It's time for lunch. Thank you to all
Thank
the
you.
speakers of the session this morning.